wasn't as bad as I feared. Could be the beer. Yeah. Yeah, no, thanks so much, Lucinda. I appreciate it. And I think I got to talk to a couple people because you got more stuff than I thought. I thought I could control it. Hey, I'll give you a few pictures. It'll be fine. But you are a good researcher. Yeah, so now I have to give a talk. And yeah, so when Mike was talking to me, Mike's always talking, right, to people, right? Kevin, have fun with it. Talk about what you want to talk about, right? So, yeah, so this is going to be an interesting talk because it's nothing I've spoken about before outside of Merck, right? So, provocative title, just to scare me. But let's start with a couple things. We're still friends? Good? Yeah? Yeah. This guy, Russell, yeah, we're still friends. It's okay. I'll get you back. Don't worry. Yeah. So that's fine. Yeah. So this is what I was thinking about when I was putting this talk together. It's like a car crash. You're going to wince, but you're not going to be able to look away, right? Yeah. And I want to thank these guys here. He wasn't in the picture. I had to stitch him in. He's a little guy, right? So, right. And Mike and these two knuckleheads on the end, right? So they just happen to be there. So this talk, we'll see how it goes. It's late. I'll zip through it. It won't bother you too much. So go back to CPSA a few years ago when we had a session on innovation. Roger Hayes was the program chair. I made the mistake of saying to Roger, "Oh, regulated BA, just shut your brain off at the door," right? And he didn't like that. And so then he made me do a session on innovation. Yeah. So, but it's an ongoing discussion, right? It's like we've outsourced a lot of bioanalytical work that we do inside of Merck, and we whinge about that. It's seen as a commodity that can be bought and sold. So really, are we innovative? Can we improve? Where can we improve? How do we promote what we do and let people know, hey, we can do some really cool stuff that you don't even think that we can do, right? A lot of things get done because that's how we've always done it, and we have to do a better job of explaining to people we can do a lot more for you, right? So are we too modest? Right? So let's jump into this, and this is a well-used slide, right? And you look at this, and from a bioanalytical perspective, hey, PK was a problem, and now it's not so much of a problem, so well, we're done, right? You look at the other areas. Well, I don't pick the targets, so I can't do anything about efficacy. Safety talks. Well, a rat isn't a human, so that's not my problem either, right? So you could say we're done from a BA perspective. I can't do anything about these, right? But I think it's like, hey, well, a pessimist, sees a difficulty in every opportunity, and a positive thinker, optimist, and I'm an optimist, sees an opportunity in all this difficulty. So, is BA a dead end or an opportunity? Well, I'll go a little step further. I think a CPSA thinker, right, sees an opportunity in this. So when you think about, hey, well, PK is a solved problem, I can't do anything about efficacy or safety, I don't think that's true, right? Because you think about it, it's data and this therapeutic index, right? So this is what we do. We measure drug levels, or we measure biomarker level, and at some dose, you'll read out efficacy, and then at some other dose that's higher, you'll get some toxicity, and the distance between these two is the therapeutic index. Well, these numbers here and these numbers here aren't points. Sometimes we like to think they're points, but there's actually variability, a spread associated with these, right? And that's where we come in. It's like understanding that spread, is what we have to do, whether it's a measurement of a biomarker, of efficacy or toxicity, or the drug level. Anything we can do to improve the measurements on these axes will help us better understand that therapeutic index. And that's what I want to talk about. Because really, it's the data that we have allows us to build models that drives programs, right? So you measure a bunch of patient samples, whether it's a biomarker or exposure, and you develop a model, right? And as this guy said, when you have bad data, you develop bad models, and then you make bad decisions, right? And he also invented the sad smiley face, right? So really, analytical data is the fundamental unit used to build models and make decisions, right? That's what I do. We generate data, right? You have to understand that data, right? So I want to talk about a few opportunities that I see for us, right? And they're not new necessarily, and they're not specific to Merck, but microsampling, so really small amounts of sample, microdosing, really small amounts of drug, and then biologics and quantitation by LCMS, so really big molecules, right? So without data, you're just another person with an opinion, right? And this is attributed to Edward Deming, but there's actually no data to support that. So, right? So a few cartoons here. So, really we've seen the advances from our innovative vendors that make instrumentation for us, and then we get to apply it. So microsampling approaches. So things like fine needle aspirate, going in and taking a really small sample from a tumor, a liver, being able to analyze that for drug levels or other compounds of interest, getting access to the samples, so collecting dried blood spot samples or capillary microsamples. The only reason we're starting to think about these is because we have good scientists and more modern instrumentation and automation that allows us to analyze these really small samples. And this gives us these new improved data streams. So we've done serial sampling and PK. That's something we did at Merck. As Lucinda said, 14 years ago now, right? And we've been using it ever since. And it was because we had, good tools to be able to do that. Measuring drug levels in liver. If you're treating a patient and you have to know what the exposure is in the liver, because that's what's really driving efficacy, how are you going to do that? And then also talk about, pop PK studies, getting data that you normally wouldn't be able to get access to by collecting, say, something like dried blood spots. And that's where, trying to build these models. You can't build the models without data, so call it, smart PKPD or smarter PKPD. It's, some of the things we've done, DBS for pop PK studies, looking at drug levels in HCV patients, and then something that we're working on, capillary microsamples for GLP type studies. So I just want to go through an example of some dried blood spot work because I think it's taken a bit of bashing, but I think there's actually a good future. So this is a study, phase 2 study, an efficacy and pop PK study. Really the goal was to facilitate dose selection for phase 3, and it was a migraine trial, right? So you're not going to ask a patient, hey, you're having a migraine, come on in, we'll take some blood from you, right? They're at home, right? And so you want to be able to collect samples at home during the event. So they have a migraine, they take your med, and a couple hours later, they'll collect a sample, right? And so what we did is plasma sampling in the clinic during a couple of visits, and then DBS sampling both in the clinic during these visits and then outpatient. And so the patient collected samples. Let's go to the next slide because this is sort of how it was done. So they come in for a screening visit. We teach them how to collect a DBS sample, and we collect a plasma sample at the same time. They go home. They have a migraine. They dose. A couple hours later, they take a dried blood spot sample. A few hours later, they take another one. And then they come back, and before they come in, they take the drug again. They get a trough sample. These are all plasma. And then we take another DBS sample. So we did that. We ran that trial. And what we found out is that DBS assay is at least as accurate as plasma. Actually, it's better than the plasma assay that had smaller residual variability. And then the data that we collected in the clinic for DBS and plasma showed that really super robust. What we did find is that DBS data collected and recorded by unsupervised outpatients showed remarkably larger variation compared with DBS, right? So basically, it was 100% variability, right? And then what are the possible sources of variation include, but not limited to, imprecise diary entry and improper collection of DBS samples. And this was like an aha moment for me because our head of PKPD basically said, oh, well, yeah, the biggest problem with our models is patient adherence. We don't know if they're taking the drug if they took the drug, And in this case, we don't know when they took the sample, right? So what do we do about that? So you think, okay, we'd walk away from DBS, but we didn't. We actually looked in other trials. So we validated several assays for DBS. This is a comparison plot of DBS versus plasma and isolinear. This is data that we took, as Lucinda said, to the EMA. We sent it to the FDA. And they're like, yeah, that looks great. And we've dropped plasma sampling in phase 3 trial for this particular compound, right? And so we're getting data, in phase 3 that is going to really drive, these late stage studies. So what we've learned, though, is we have to improve how we collect these samples at home and the information that supports it. So we're doing things like e-diary and testing e-diary technology, anything to improve patient compliance. And I'll talk about that at the end after I get through these examples. This smarter PK/PD, right? So this is what we have to develop, and you talked about it, the pre-analytical variability. And so, okay, so that's small samples. What about small doses? So microdosing, been around for a while. We've done it. Here's an example from a paper published by Lee Sun at Merck, where we dosed 4 compounds in clinic at phase 0, measured by LCMS because we couldn't really predict the human PK. You can do absolute bioavailability, where you put a labeled compound, whether it's C14 labeled or stable isotope labeled, as an IV dose on top of an oral dose, and you get absolute bioavailability. But what we're doing that's kind of interesting is looking at supporting DDI studies, where we're developing panels of compounds that we can microdose and allow us to get information about our compounds as perpetrators of drug-drug interactions. So these new data streams are used to improve decisions in the clinic, right? And so some you can do by LCMS, some you have to do by accelerator mass spectrometry. But this example here, this slide from Cynthia Chavazang, who, when I was leading the methods development groups, worked on this project. It's a series of statins, right? And these compounds are substrates for transporters. But when you give them at a higher dose, a therapeutic dose, it gets confounded in terms of which transporter is involved. You might have a saturation of a certain pathway, so that's why you microdose. You go down really low so that their PK is not interfering with each other. And then when you dose your Merck compound or your compound, you can start to see, if you're starting to perturb the PK of these compounds when they're microdosed, you'll start to understand whether or not you're having an effect on transporters. And so to do that, you got to dose really low, so tens of micrograms, 20 micrograms, 30 micrograms, which is fine, and we can dose that, but then you got to be able to measure them. So, the advanced analytical technology that we have available to us and people in the lab that can develop the methods allow us now to get down to this is 1 to 1,000 picogram per mil, right? So but 1,000-fold less than we'd normally do for an assay. Instead of nanogram per mil, it's low picogram per mil. So again, it's these data streams that is helping us drive better decisions in the clinic. And then the other topic around protein quantitation. So obviously very important, very popular, but we come at it from a different perspective, right? So there's the biomarker side of things, but we work in drug metabolism, so there's ADME proteins, right? And understanding ADME proteins is important because for pediatrics, for example, little kids just aren't little adults. The expression of ADME proteins changes as you age. And so when you're trying to scale a dose down to a child, you have to understand what the expression level of P450s, for example, are in a child, right? And so getting access to liver samples, it's unfortunate they're available, but they are available. And so you can go in and start understanding what the expression levels of ADME proteins are in pediatrics. Disease state has an impact too, right? So understanding what the expression levels of transporters or P450s or UGTs are in diseased livers helps us build better models to advance our clinical compounds. And the other side then is quantitating proteins that you actually put into people as patients, so therapeutic antibodies, right? So ligand binding versus mass spectrometry. Really what it's about is getting better data so you can build better models, right? So an example here, right? So this is stuff that we've been doing recently on quantitating antibodies by LCMS, right? So we're looking at ligand binding data versus LCMS data, and the green line is LCMS data from a 10 mpk dose, and then the same dose by ligand binding is down here. So why is this higher, right? Why are they different? How do we interpret the data? Which one is right, right? Well, they're both right. They're just measuring different things. You have to understand the format of the assay. And, you know, if you're a toxicologist, say, hey, I want the higher line because that's total, you know, body burden. I'm seeing higher exposure. But if the lower measurement is related to the portion of the protein that is efficacious, then that becomes important for doing your PK/PD modeling, right? What we can do, though, is then do other things that you can't necessarily do with a ligand binding assay. So in this case, the PCSK9 ligand is circulating. You put an anti-PCSK9 antibody in the patient, and then you pull it down. Well, you pull the ligand down with it. We're doing digestion, and so you digest that PCSK9 ligand, and then you can monitor its peptide as well. So you can track how much PCSK9 is bound to that antibody. So if you're stopping up a soluble ligand, you can actually measure that. So now you could develop a ligand binding assay against the ligand as well and do that. So that's, you know, okay, that's good. We can do it. And you know, we've recently published, had a postdoc working in the group doing that. Dan Spellman's been really instrumental in getting this up and running as well. But we can go beyond ligand binding assays and look at things like glycosylated proteins. So Baogen has done a lot of work on developing an assay that was originally set up in our Palo Alto group, and we've transferred it over. And it's really looking at these replacement therapeutic for using intravenous immunoglobulins. And so what they do is they take a big pool of immunoglobulins, combine them together, and then they dose that to people. And the theory is, based on this paper, that the extent of glycosylation is what drives efficacy. Now, the ligand binding people develop an assay and can measure total protein, but they can't pick up the extent of glycosylation. And that's where LCMS comes in. So we set up an assay, we automate it using Agilent assay map system, get a nice chromatography and sensitivity using our ion key on our triple quad, and we can get the exposure, but now we can also, of the total protein, but we can also go in and measure the glycoforms as well. So now we get an understanding of the extent of glycosylation that drives efficacy. So this is stuff that you just can't do unless you're developing advanced analytical technologies, right? And this is the type of thing that we have to bring to the project teams, because they'll, in this project team, they're like, oh, well, I can only do ligand binding, I can only measure total protein, and then it's like, hey, no, look, we can actually do something for you here. So we got the opportunity, and now this data is being helpful in understanding the animal models and the exposure related to efficacy. So three different data streams: microsamples, microdosing, proteins. What do we do with these new data streams? What's next? So that's the bioanalyst dilemma, right? So we're outsourcing stuff. Anyone can buy a mass spec. Hey, that's really cheap, says our procurement group. So-and-so bought an instrument. So, if you think it's expensive to hire a professional, wait until you hire an amateur, right? Right? And I don't know. Many of us have probably heard this, right? Are you sure that data's right? Can you rerun those samples? That's not what we expected. Did you mix something up, right? So it's like, I did your analysis with your bad assumptions, then I applied your flawed logic. Should I begin disillusioning the team? Well, really, add a pie chart, right? So variability has many sources, and it's not just from the analysis of samples. And as we build these new data streams, they're new data streams, right? We haven't had them before, so we have to understand them. All right, so bioanalytical scientists need to look for opportunities to bring these new data sets to project teams. It's really promote what we can do that's novel and new, and because people on project teams don't know, right? They don't know what we can do, right? And unless we're there telling them, then we're not, they're not going to change how they do things, right? So really, these new data types need to be understood and integrated. Data to knowledge to understanding. And really, we have to understand the sources of variability in these new approaches versus old approaches, because we'll always be held to, hey, well, I always do it this way. This is my gold standard. Can you beat it? And the old method may actually not be the most appropriate approach. So, yeah, pre-analytical. It was good. I was putting this talk together. I was thinking about this, and then all week we've had some really good talks talking about the problems that go beyond the analytical aspect. Running a mass spec is easy, so I've been told, right? But all the aspects that go into getting your sample. So pre-analytical, there's lots of sources of error there. Post-analytical, your model could be wrong. So what are the sources and impact of variability in clinical trials? So that's the last part. So smart PK/PD. So we're close to going to the bar. You'll be happy to know, right? So here's concentration, time. And this is like a multi-dose study, and this is PK profile, right? So it looks okay, but then you get these big dropouts and these spikes. It's like, what the hell's going on? What did you do wrong? You didn't analyze these right. Well, this data is from a website where they're promoting adherence technologies. And in this case, they were actually measuring adherence. And so these black bars are when this person didn't take their med, right? So no surprise, no level, no med. Double dose, double spike, right? So, how do we understand, if the biggest source of variability in clinical trials is not knowing whether someone took their drug, well, we should do something about that, right? This is a really compelling story. This is HIV treatment, right? And this colored in area is the adherence. And then the point is the efficacy and the confidence center around it. So low adherence, low efficacy, high adherence, high efficacy, right? So that's important, right? And so from a bioanalytical perspective, what can I do about that? Well, if you're not taking the drugs, they don't work, right? So drugs only work if people take them. And so this data is from this paper. It's a really good paper. I think you should read it. Clinical Pharmacology and Therapeutics. That's just this year. So drugs don't work if you don't take them. You can't build a good model if you don't have data, right? And that's our job, to inform people. Hey, we can generate new data streams for you. But also we have to go beyond that and have tools to improve. And I talked about that at-home sampling for DBS. Well, we have to build better tools to collect those samples at home, right? And that's really going to drive future clinical development plans. So this is an interesting plot. It's adherence and efficacy, right? So phase 1, it's easy to get good adherence because you have the person in the study locked down, giving them, the drug. But as you go through later trials, the adherence level drops off, and then you see a drop in efficacy. Well, you could kill a whole program because you think that you don't have efficacy when, in fact, it's really an adherence problem. And then when you go out into the real world, it drops even further because it's in a bigger patient population. So this is, personalized medicine, right? We talk about it a lot, but to me, this is what I think about is personalized medicine, finding the right drug for the right person so that this use effectiveness is actually higher because you're making sure the person has the right drug. So smart PK/PD, right? Tracking if and when patients dose. So smart packaging and smart pills, accessing samples beyond the clinic, sample collecting devices, remote monitoring of patients, things like wearables, and then how do we integrate this all together? So this is what it excites me now. This is what I think about a lot. My boss here, Jane Harrelson, is really helping to drive this initiative at Merck. So blister packs that have RFID chips in them, so when you open it, it sends a signal to your smartphone, and so you can track when someone took it. Well, it doesn't say if they took their medicine. It just says they opened a package. So we go beyond that, and so Proteus Health published a paper on these edible sensors, right? So that looks kind of, not fit for prime time. You got to wear this thing on it. But this is the paper. This is the reality now. It exists. It's an FDA-approved device. So it's an RFID that goes on a pill, gets activated by your gastric juice, sends a signal to basically a patch that you wear. So you can monitor, in real time whether someone took their med or not, and when they took it. And then blood collection. We've seen a lot of that this week. So there's different companies out there that have different devices. So Spot On Sciences, Phenomenex has their new device based on this polymer that absorbs a certain amount of liquid. Novolytics, we've seen this week. Now they're advancing it and being able to collect more. This company that has, again, a separator device, they send it home and you can collect blood at home and they'll do analysis for you. And then this, 7 Sense Bio, a different approach to collecting. And there's actually going to be a talk tomorrow. I don't know if Tim was supposed to fly in tonight. I don't know. I've never met Tim maybe he's not here. He'll be here tomorrow. Anyways, a couple slides on this. So this gets away from the lancet, so instead of stabbing your finger and bleeding, this is a microneedle-based device that is supposedly painless. I haven't tried it myself. So you put it on, and you have microneedles. There's a vacuum that lifts up the skin, and then the needles puncture the skin, and you collect a sample, all in that device. And Tim's going to talk about it tomorrow. But this opens up a different approach to collecting samples. They're going to build in date and timestamp type collection information. They want to go beyond that and build tests actually into the device. And then wearables. So different companies are making things that you could wear that monitor your vitals, iWatch, and then glucose monitoring, adapter for your phone, Google contact lens that does monitoring of your glucose levels. So these types of things are coming. Data streams that become really powerful to better understand our clinical trials. But we have to bring it all together, right? So these new sample types, home-based sampling for PK, biomarkers, we need to make sure that we're collecting them in a way that we know when they're collected. And then we have to build the models associated with that and bring all these together. And there's companies out there that are thinking along these lines. So the Wi-Fi enabled, patient sensor, whether it's at home or in the hospital, and bringing all these data streams together so you get real-time monitoring of a patient. So this is at home, but you can imagine how this could be used in the clinic and allow you to get the information that you need to ensure that the data that you're collecting is actually useful, right? So a couple final thoughts. So really understanding, human biology will improve as we collect better data sets. And that's really on us. It's the bioanalytical tools that allow us to do that. So it's really about improving this efficacy-toxicity ratio are the goals. We need to collaborate across many disciplines to solve these, but bioanalysis, to me, has to be an equal partner, not a support function. We have to be there discussing what the program needs and bring our knowledge to the table so that we can access samples and do analyses that the teams may not be aware of. And then reducing variability, or at least understanding it, needs to be part of the solution. So access to these new data streams and improving adherence, right? And so these technologies are coming, and the bioanalytical scientists need to lead the way. And that's, so this is, stuff that we're thinking about inside of Merck, keeping us busy. And I didn't want to, so I think that I have one last slide about acknowledgements before. So I hope you understand the talk. It's something that I haven't talked about outside of Merck before. It's something that we're doing inside of Merck, and other companies are doing it. I didn't want to talk, I think, Lucinda, thanks for covering my career. I didn't want to talk about stuff that I would done. I hope this sort of spurs you guys on to think about new data streams and new ways of doing things. I think it's a big opportunity. So last, I want to acknowledge some people that have helped me over the years, helped me get where I am. So Rick King, good friend, my science god, super whatever guy, right? Super genius, right? And Laird, so listen to talk about Laird and Russell and Jose, really three guys that, good friends and keep me balanced, right? Tell me when I'm getting out of line. And Rick, Rick's a good guy to bounce ideas off because he'll say, "No, that's stupid," right? And it's like, I'm not afraid, right? So, Jim, he hired me at Merck. Good mentor. Will yell at lots of things, and right. And as long as you understand that he's yelling at things and not at you, then you're fine, right? And right. Tom Bailey, so Tom, he pushed me into a job that I didn't want, right? I didn't want, being this network leader job, but he was right. It was the right place for me. So, and then Lucinda and Prajakti, so Prajakti was running the EMA selfie. It's like, these are my work wives, right? These, right, right. They, they're happy to listen. They help me keep me again in line at work. Nancy and Jane, they're my last two managers. Jane's my current manager. Nancy and these guys are great because they give me enough rope to hang myself, but then tell me, Kevin, you're about to hang yourself, right? right. And Lisa is our VP, and so super supportive to me over the years, and I really appreciate that. Mike's been a good friend, I think since '98 when I first met him, and great guy. Mike, my psychotherapist, I think, right? I think he's everyone's, right? He goes to, right? And so my two daughters, right, Allison and Melanie, these are the guys that keep me going, right? Well, I want to be successful because I want them to see what you can be successful, right? So, I want to motivate them to be successful, so I want to be successful. And then, my wife. So this picture I put in, she's support, right? She's the one that gave up a lot for me. I wouldn't be able to do what I do if she hadn't made a lot of sacrifices. So I probably don't tell her that enough, but don't say I said it, because then, she'll just, right? But so Diane, really appreciate her support over the years. So thank you guys for listening, a little bit of my story and what I want to do. Lucinda, again, thanks very much. And, one last slide, right? This is my plea to you guys, right?
Kevin, I hope this wasn't as bad as you thought it was going to be. But it's so well deserved. Your passion for pharmaceutical science, analytical chemistry, and your humor is really it's enviable. And getting to know you over these years at CPSA has been a real honor, and I find it a special honor to be program chair together with Paul the year that you've been selected to be the distinguished analytical scientist. So once again, congratulations. All right, and that concludes this evening's events. We'll keep the celebration going.